All right, hi everybody. Dr. Jemison here again with another video lecture, and today we're going to break this up in a couple different parts. So part one is going to be normal structure and function of the hematologic system. We're going to look at blood and the components within blood and what makes those components unique. And so the first slide that I want to start with is this one right here that you can see, so make sure you're following along in your notes, and let's get to it. Let me transition over here, so now hopefully you can see my laser pointer cursor. And when we're talking about blood, I really want you to think about blood itself as being a solution. So it's a water-based solution that has a lot of different components to it that are all mixed together. And so we've got the diagram here showing us that there's 45% of the blood solution is made up of formed elements. And those formed elements are erythrocytes, which are red blood cells, leukocytes, white blood cells, and platelets. And then the rest is plasma. It makes up more than half of the blood solution. And most of the plasma component is water. And dissolved in that water, we have some solutes like some ions and salts and nutrients and things like that. But also we do have some proteins that are part of the plasma aspect of the blood solution. So these proteins, albumins, globulins, fibrinogen. So plasma has proteins. These are single proteins. They're not cells. They're proteins on their own. But then the formed elements are comprised of the red blood cells, white blood cells, and platelets. All right, so when you're thinking about blood, I really want you to think about what the function of blood is. So I've listed out 4 functions here, but there's more than that. Maybe you can think of 1 or 2 more. So the first function obviously is to deliver nutrients and oxygen to the rest of the body so that cells can live, and so that cells can use oxygen and glucose, for instance, to maintain their cellular processes. And then at the same time, another job that blood has is to remove metabolic wastes and deliver those wastes to the liver or to the kidneys or whatever so that we can get rid of those waste products. Also, don't forget how the blood participates in the immune system. Blood has white blood cells within it, and that aids in the defense against microorganisms and injury with the clotting cascade and the inflammatory response and so on. And then also, like what we just went over a couple weeks ago, blood also participates in the maintenance of acid-base balance. Can you think of anything else that blood does? One thing that it should be included on this list is thermoregulation. Blood helps us, the body, to regulate normal body temperature and disperse that body temperature as needed. Whoops. So let's look at the composition of blood. So blood, about 6 liters. The average adult is carrying around about 6 liters of blood. And again, it's made up of plasma, which is mostly water-based, and then the dissolved proteins in the plasma that we call plasma proteins, the solutes, which are electrolytes and other things that are dissolved, and then finally the cells and the formed elements. Plasma itself is basically the fluid component of blood. So I've got here 90% water. I know in the picture earlier on, just a few slides ago, it said 92%. I'm not really nitpicky about the percentages here. Just that you realize that plasma is mostly made up of water, and the blood solution, more than half of it is plasma, so it's a water-based solution. And dissolved within that plasma, we've got organic proteins and inorganic elements like electrolytes and so on. So let's look at the plasma proteins. These are the albumins and the globulins and the clotting factors. So albumin is part of the albumin family, and there are several different types of albumins, and they function as carrier proteins. And also remember, albumin is the most abundant plasma protein, so it's going to exert the most plasma oncotic pressure. So as far as the carrier protein function, we have hormones, for instance, that are not water soluble, that need to be bound to a water soluble protein like albumin in order for that hormone to be delivered throughout the body, and so on. So we have several different classes of carrier albumin proteins. And then the globulins, looking at the immunoglobulins, basically the antibodies. And then clotting factors, we'll get to this towards the end of the lecture, mostly fibrinogen, but also we have all of the other inactive clotting factors that are floating around in the bloodstream that comprise the plasma compartment of the blood. So let's look at the formed elements. Cells, defining all of these as cells might be a little bit misleading. So let's look at this. Erythrocytes. That's basically the fancy word for saying red blood cells. Leukocytes, again, that's just the general term we use for white blood cells. And then thrombocytes. Thrombocytes is another term for platelets. And thrombocytes and platelets, they're not actually cells. They're cellular fragments. And so using the term formed elements is a little bit more specific when we're talking about the cells or cell fragments that comprise this component of blood. So let's look at red blood cells first, erythrocytes. Check out this number. There are estimated to be in an average adult body 3.72 times 10 to the 13 red blood cells at any given time. That's nearly 37 trillion red blood cells. That's why red blood cells are the most abundant cell in the body. We've got so many red blood cells. And their basic job is to carry oxygen and deliver that oxygen to the cells of the rest of the body. And the red blood cells have a unique morphology in that we can describe them as biconcave discs that have reversible deformity, which means that they're flexible. And the biconcavity means that they're not spherical, they're more flat, kind of pancake in shape. And red blood cell normally lives about 120 days, and then it's all used up and the components of it get recycled. So looking at this scanning electron micrograph, we've got a red blood cell here right in the center, and we can see this biconcave disc. So it's kind of flat, pancake-ish, but it has this divot on either side of it. And basically what that allows for is maximum surface area of the red blood cell membrane to allow for maximum gas exchange to occur, because red blood cells are basically little packets full of hemoglobin, and hemoglobin's job is to bind and release oxygen and to a certain extent CO2. And so if we can maximize surface area without overdoing it on the volume size of these cells, it makes it the most efficient morphology for this function. So also in this picture we see these white blood cells here, kind of fuzzy looking, and then these green items here, these are platelets, thrombocytes. So we'll talk about these coming up in the next few minutes. Let's look at the leukocytes, the white blood cells. Most of this should be reviewed from our immunology section, and basically what white blood cells are doing is getting rid of waste products and protecting the body against foreign invaders, pathogens, antigens, and so on. So we can classify white blood cells into different types based on what their function is and what they look like. So the first group we have are called the granulocytes. And they're called granulocytes because it looks like there's granules or little bits of sand or chunks within the cytoplasm, but basically these granules are packets full of enzymes and other things that help that white blood cell do its job. And we do have granulocytes that participate in the inflammatory system as well. Do you remember what the granulocytes are? Here's the acronym for you, BEN. So BEN is the granulocytes, so basophils, eosinophils, and neutrophils. All of the other white blood cells and lymphocytes would be considered agranulocytes. The prefix A meaning that these are without granules within their cytoplasm. So let's look at the granulocytes first. So basophils, eosinophils, and neutrophils. Neutrophils, remember, they're phagocytic cells that show up in the early inflammatory process. Once neutrophils leave the bloodstream, they don't return back to the bloodstream and they don't go into the lymphatics. They just go out, phagocytize, and then their job is done. Eosinophils are related to hypersensitivities, so they interact with IgE and IgE-based hypersensitivities. So do you remember which type of hypersensitivity IgE is related to or participates in? If you're thinking type 1 hypersensitivities, you're right. And then also eosinophils participate in parasitic infection control as well. So most of the time here in the US and in industrialized nations, we see eosinophils participating more often in inflammatory and hypersensitivity reactions and less often for parasite infections. Also, eosinophils, they play a minor role. They have a minor phagocytic activity. I'm not too concerned that you keep that in mind. Mostly just the hypersensitivity inflammatory concept or characteristics of eosinophils and parasitic infections. And then finally, the last granulocyte are the basophils. And remember, basophils are found in the blood. Once those white blood cells move out of the blood supply and into the tissues, they become mast cells. And we've got mast cells everywhere where there's vascularized connective tissue. So mucosal surfaces, respiratory tract, GI tract, capillaries of the skin, and so on. So basically we've got mast cells everywhere. And then apparently there's a controversy as to whether or not mast cells are true white blood cells or not. For me, I consider mast cells white blood cells because they come from basophils, which are definitely a white blood cell. So if you ask me, there is no controversy. Mast cells are white blood cells. And then we've got the agranulocytes. So these are the white blood cells that don't have any granules in their cytoplasm. Macrophages and monocytes are part of the agranulocyte group. And throughout this lecture, coming up in just a couple more slides, I'm going to mention the MPS, or the Mononuclear Phagocyte system. This is a system of organs and specialized macrophages that will clean out debris from the blood and help to clean up pathogens that might have found their way into the blood supply as well. So we'll get there. Put that on the back burner for right now. And then finally, lymphocytes. Remember, the lymphocytes are the T cells, the B cells, and the natural killer cells. Lymphocytes are still considered agranulocytes. And then lastly, thrombocytes, which are the platelets. And remember, these are not cells themselves. These are cytoplasmic fragments from a much larger cell. And what platelets' main role is to participate in the blood clotting cascade of events, which controls bleeding and prevents loss of an excessive amount of blood. So then how are platelets or thrombocytes made? Well, there's a hormone called thrombopoietin, which is produced by the liver mostly, and the liver will make and secrete this hormone, thrombopoietin, and the target tissue of this hormone is bone marrow. And when the bone marrow cells get stimulated by thrombopoietin, then that will stimulate those bone marrow stem cells to produce more platelets. So we'll look at that process also coming up. But as far as right now, thrombopoietin is the hormone that stimulates the production of platelets in the bone marrow, and thrombopoietin is made by the liver. Here's a pretty good picture of a platelet, and this platelet has been activated, and we know this because inactive platelets are kind of spherical or round, and they don't have these projections. They don't have these extensions. When a platelet becomes activated, then it will send out these projections and start grabbing onto and sticking to whatever's nearby. And that's the characteristic of an activated platelet. I always thought that this picture looks like a goat head thorn. If anybody's familiar with goat head thorns, you might know that if you step on those with bare feet, they hurt really bad. But also, if you ride your bike over a goat head thorn patch, then you're bound to get flat tires because the thorns actually look like this, and they have those strong projections, needle-like projections that will just poke right through your tire and puncture that tube. Okay, so let's look at the lymphoid organs. Specifically, let's look at the spleen and lymph nodes. Spleen, largest secondary lymphoid organ. It sits there next to the stomach, kind of posterior lateral to the greater curvature of the stomach. And think of the spleen as a giant blood filter. And within this filter that we call the spleen, we have macrophages that live in there, we've got T cells that live in there, we've got B cells that live in there, and also somewhat we've got some red blood cells that are kind of in storage there as well. And what the spleen's main job is to filter out pathogens and junk from the blood, but also to get rid of old and damaged or dying red blood cells. And then the spleen will break those red blood cells down and recycle their components. So here's another scanning electron micrograph of spleen tissue. So what do you think these cells are that are numbered 1, 2, 3, 4, 5, and 6? They're all the same kind of cell. So I'll give you a second. Okay, time's up. These are red blood cells. And if you notice, they don't look like that typical biconcave disc that we see in these other pictures. That's because, remember, red blood cells are flexible. And when they're filtering through the spleen, they have to go through these really tight openings. And that allows for the macrophages and other white blood cells within the spleen to really get a close look at these red blood cells. And if the red blood cells are damaged, or dying, or actually not working anymore, then the macrophages can start destroying those damaged red blood cells. So they're squeezing through these little openings so that they can be closely monitored. So these cells right here, maybe this one right here too, these are white blood cells, maybe macrophages, I'm not sure. And then these smaller roundish looking structures labeled P, you might be guessing platelets, and you would be right. So they labeled it P for platelets. And then the arrow here is just pointing at one of these cytoplasmic extensions off of a red blood cell. Apparently that was interesting to whoever published this picture. Okay, so then lymph nodes. I know we've talked about lymph nodes a little bit in previous lectures, but let's just review. Lymph nodes are definitely part of the immune system because, remember, T cells and B cells live within the lymph nodes, and those white blood cells, those lymphocytes, are monitoring all of the lymphatic fluid that's being filtered through the lymph nodes. And so that's why we can say that lymph nodes help to cleanse lymphatic fluid of microorganisms or pathogens or particulates, especially if they're foreign particulates, and so on. So let's look at a close-up view of a lymph node, and we have several afferent lymphatic vessels bringing lymphatic fluid into this lymph node, and we have all of these areas of T and B cells that survey and monitor what's inside of that lymphatic fluid, and then finally the lymph node is drained by a single efferent lymphatic vessel. So if there's an antigen or a pathogen that arrives to the lymph node, then we might have clonal selection occurring where a particular T or B cell recognizes that antigen, and then we've got the clonal expansion and all of that immunological stuff that we talked about earlier in the quarter. Okay, now let's shift gears a little bit and revisit this mononuclear phagocyte system, or the MPS. Remember, this system is comprised of specialized macrophages that are found mostly in the liver and spleen. So I'm not saying that all macrophages are mostly found in the liver and spleen. I'm saying that in the MPS we've got specialized macrophages that reside in the liver and spleen. And basically what those macrophages are doing are looking for microorganisms or damaged red blood cells or what have you, foreign material, and then they will phagocytize those things and destroy them. So that's one of the functions of the spleen, but also the liver participates in that function as well. And these two organs, with their specialized macrophages, comprise what we call the MPS, or mononuclear phagocyte system. Okay, so how do blood cells, not just red blood cells, but all blood cells form? Well, we call that process of blood cell formation or production hematopoiesis. And with each blood cell, hematopoiesis basically occurs in two basic stages. So they all start out as a stem cell, and then the stem cell will divide through the process of mitosis and continue to divide. And with each division, that new cell will mature and differentiate more and more into the blood cells that we're familiar with, whether it's red blood cells or a granulocyte or an agranulocyte, and so on. So the word pluripotent here, as far as pluripotent stem cells, this basically means that we've got stem cells within the bone marrow that have multiple potentials to become any type of blood cell, whether it's a red blood cell or a white blood cell, and so on. So pluri means multiple, and then potent, just think potential. So depending on the cytokines that are stimulating the pluripotent stem cell, or maybe even hormones that are stimulating these pluripotent stem cells, will determine what mature cell that pluripotent stem cell will end up as. So as you're reading and studying about this process, you'll come across the word multipotent and also totipotent stem cells. Pluripotent, multipotent, and totipotent, they're all describing the same type of stem cell. So this earliest form of stem cell that, depending on what kind of influence it's being stimulated by, whether it's cytokines or hormones, will have the potential to become any type of blood cell. So let's look at basically a family tree of blood cells. So up here at the top, we've got, it's almost totally cut off, but it says pluripotent stem cell up here at the top. So this is in the bone marrow. This stem cell could become any number of blood cells. So it could become a red blood cell, could become a eosinophil, could become a T cell. So given the right cytokines and hormones, this pluripotent stem cell will change into a common myeloid stem cell or a common lymphoid stem cell. So once we can identify a common myeloid or a common lymphoid stem cell, then these stem cells are kind of committed to become a myeloid cell or a lymphoid cell eventually. So you don't have to memorize all of these in-between stages of blood cells, but what I do want you to recognize is what a pluripotent stem cell is, or totipotent or multipotent, it's all the same thing. And then given the right circumstances, that pluripotent stem cell will become a common lymphoid stem cell. And once we see a lymphoid stem cell, this stem cell is destined to become one of these lymphocytes. So if you're worried about memorizing these characteristics, all you have to do is remember that the lymphoid stem cell gives rise to the lymphocytes. And there's only 3 lymphocytes: B cells, T cells, and natural killer cells. All of the rest of the blood cells come from the myeloid stem cell. So this myeloid stem cell can eventually become an eosinophil or a monocyte, neutrophil, megakaryocyte, or a red blood cell or basophil, so on. So that's a really easy way to remember that. And this will make more sense when we talk about the hematologic pathologies, especially with the leukemias and lymphomas. So here's the the family tree from your book. I'm not sure where this other one came from. But here's the image from your book. So we've got this hematopoietic stem cell, which is basically the multipotent stem cell within the bone marrow. And then given the right stimulus and conditions, that stem cell can become a common lymphoid stem cell, which will give rise to the lymphocytes, or it'll become a common myeloid stem cell, which will give rise to all of the other blood cells that we find within the blood. So again, don't get too caught up, and I would say don't even worry about learning these intermediate cells along the maturation pathway here. Just know that myeloid stem cell gives rise to all the blood cells that are not lymphocytes, and the lymphoid stem cell gives rise to the lymphocytes, so the B cells, T cells, and natural killer cells. All right, so hematopoiesis occurs in the bone marrow. That's where we have those stem cells. And an older term for bone marrow is myeloid tissue. So if you might come across that terminology referring to bone marrow as well. We've got red and yellow bone marrow, and I know you guys can read this, so I'm not going to highlight too much of this, but in adults we do have active bone marrow because we're constantly producing new white blood cells and new red blood cells, and here are the areas where we have active bone marrow in the adult body. In looking at the cells that are produced in the bone marrow, we've got the stem cells that are found in the bone marrow. So here it is, multi-potential or totipotential, pluripotent, all the same thing. So this stem cell could either become a common myeloid stem cell or a common lymphoid stem cell, and then give rise to those respective blood cells. So if we're looking at the granulocytes, 50% of the granulocytes are found stored in the bone marrow, and then 50% are out in the periphery doing their job. So we've got a reserve of granulocytes. In case the ones out in the bloodstream get exhausted or all used up, we've got some mature granulocytes already stored in the bone marrow, ready to go. Thrombocytes, on the other hand, we don't store very many of those in the bone marrow. And then red blood cells, we don't store any mature red blood cells in the bone marrow. Once a red blood cell is formed and totally mature, it's sent out to do its job in the bloodstream, and there's no reason to store those red blood cells because we're constantly making new ones. Remember, their life cycle only lasts about 120 days. So let's look at red blood cells, and specifically how red blood cells are formed and what stimulates their formation. So the process of forming red blood cells is called erythropoiesis, and same process as hematopoiesis, where we've got stem cells in the bone marrow that are stimulated by, in this case, erythropoietin, which is a hormone that will stimulate bone marrow cells, stem cells, to produce red blood cells. And every time a baby red blood cell or a developing red blood cell divides and matures, the nucleus will decrease and hemoglobin concentration and content will increase, until finally, when we have a mature red blood cell, there is no nucleus in those cells. All they are packed full of hemoglobin, so that they can maximize their job as delivering oxygen and taking CO2 back to the lungs. So let's look at erythropoietin here for a second. This is the hormone that stimulates red blood cell formation. So here's where I like to ask people if they know where erythropoietin is produced. Where is it made? And usually the liver is the right answer for these types of questions, but this time it's not. Erythropoietin is made by the kidneys, and we'll look at that in just a moment. So thrombopoietin is produced by the liver that will stimulate bone marrow to make more thrombocytes. Erythropoietin is produced by the kidneys that will then stimulate the bone marrow to produce more red blood cells. We usually abbreviate erythropoietin and thrombopoietin. Erythropoietin, we usually abbreviate it as EPO, and thrombopoietin as TPO. So you might see it both ways. So up here in the green box of this flow chart gives us scenarios that might stimulate the production of more red blood cells. So let's look at a few of these. Decreased population of mature red blood cells. So if there's not enough mature red blood cells, then there's not going to be enough oxygen carrying capacity of the blood, and that causes problems. Hemorrhage. Again, we're losing red blood cells, so oxygen carrying capacity of the blood diminishes, and so on. So if we don't have enough red blood cells, or if the tissues of the body require more oxygen than what the current population of red blood cells can provide, then that will stimulate specialized cells within the kidneys to secrete EPO, erythropoietin. And so the kidneys are the ones that are monitoring whether or not there's enough oxygen concentration in the blood, but also whether or not there's enough red blood cells and hemoglobin concentration in the blood. And if there's not, then these specialized kidney cells will produce and secrete EPO, and then EPO will travel to the bone marrow to stimulate the formation of more red blood cells. And then we get more quantity of red blood cells being produced, and that increases oxygen carrying capacity within the blood. And once that is increased, then those specialized kidney cells are satisfied, and so they'll stop producing as much EPO. So here's kind of the stage of events for producing a red blood cell. So we've got this pluripotent stem cell, again, found in the bone marrow. But once that stem cell gets stimulated by EPO, erythropoietin, then it is committed to become a red blood cell. And with every cell division and every stage of maturation, the nucleus of this developing red blood cell gets smaller and smaller, but hemoglobin concentration increases more and more until finally we've got this mature red blood cell that has maximum hemoglobin and maximum oxygen carrying capacity abilities. So again, regulation of erythropoiesis really depends on how much oxygenated blood flow is getting to the kidneys. So if there's hypoxia or hypoxemia, both situations, that will stimulate the production of EPO by the kidney cells. And as far as knowing the peritubular cells of the kidneys, I'm not too concerned about that at this point, but when we do talk about the renal system coming up at the end of the quarter, we're going to revisit which cells are producing EPO. So this is not the last time you're going to hear about erythropoietin. Something that's interesting to think about is when you hear about athletes that are blood doping, there's a couple different things that are going on with this. So blood doping could mean that the athlete is injecting exogenous erythropoietin to stimulate their bone marrow to make even more red blood cells in the absence of hypoxia or hypoxemia. Or athletes will transfuse their own packed red blood cells that they donated to themselves a while ago, and then now they're reintroducing that into the bloodstream to increase red blood cell population. And what that does is, if we have more and more red blood cells, we've got more and more oxygen carrying capacity in the blood, and that allows for muscles to maintain high oxygen requirements for a longer amount of time. And so that's why it's considered cheating in the world of sports. But there is a way around this where athletes can naturally increase their red blood cell concentration, and that happens quite a bit here, especially in our state, Colorado. Athletes from around the world will come to Colorado to train because we have high altitude areas, especially up in the mountains. We've got training centers up there where the athletes will be at a higher altitude, exercising, training, and the higher in altitude you get, the less oxygen concentration there is in the atmosphere. So then that will stimulate EPO naturally to be produced by the kidneys and stimulate more red blood cell production. So then once the athletes are done training, they'll go back down to sea level or lower altitude to compete, and they already have extra red blood cells produced. So just something interesting to think about. And in fact, there are certain masks that people can train in so they don't have to travel all the way up to the mountains. High altitude masks that kind of reduce the oxygen concentration while athletes are training, and that just stimulates EPO production as well. All right, so looking at red blood cells in particular, I keep saying that they're little cellular bags full of hemoglobin, and that's basically what they are. So each red blood cell contains as many as 300 to 400 million hemoglobin molecules within that single red blood cell. So anybody who really loves math, maybe you can do the math for us. If the average adult has about 37 trillion red blood cells, multiply that by, I don't know, let's say 300 million, we can calculate how many hemoglobin molecules there are in the human body. We could take it even further and get even more nerdy with this. So how many iron molecules are found in a single hemoglobin molecule? If you remember your anatomy and physiology, there's four iron molecules per hemoglobin molecule. And so then you can divide that total number by 4, and we can calculate how much iron is carried around and used by the average adult human. So anyway, as far as hemoglobin goes, just remember that there's 2 pairs of protein subunits, the beta chains and the alpha chains. I'm not going to ask you what the difference is between these. You guys have already done this in anatomy and physiology. But what I do want you to recognize is that there are 4 heme groups within a hemoglobin complex, and that's where the iron is found, is in these heme discs. And the iron is there as the oxygen binder. So each hemoglobin complex can carry around at most 4 oxygen. So when there's not oxygen bound to hemoglobin, it kind of takes on this bluish color, and when there is oxygen bound to hemoglobin, it takes on this kind of reddish color. So that's kind of the difference of why we see different colors of blood, whether it's in the venous system or the arterial system. It just depends on how oxygenated those hemoglobin complexes are. Something that I'm going to introduce right here and not really talk a whole lot about is the nutritional requirements that are needed for hemoglobin synthesis. So obviously we need proteins and amino acids. Those are what the alpha chains and beta chains are made up of. We also need a number of vitamins to support the enzymes that are producing hemoglobin, and minerals as well. We need iron, obviously, and copper participates in some of this functionality as well, and folate, folic acid. So nutrient depleted folks, we would expect to see less effective and maybe even less amount of hemoglobin production in these nutrient deprived individuals. So once a red blood cell reaches the end of its lifespan, remember it's about 120 days, then the macrophages of the MPS, mostly in the spleen, will destroy those red blood cells and break them down into their components so that the body can recycle those components. So the globin chains, because they're protein chains, they're made up of amino acids. So when red blood cells need to be recycled, these globin chains get broken down into amino acids, and then that frees up those amino acids to become other proteins. The heme discs, or porphyrin rings, get metabolized into a substance that we call bilirubin, and then bilirubin gets transported to the liver and the kidneys and gets excreted either by way of the urine or the bile. And then just as another reminder here, we need adequate amounts of B12 and folic acid in order to maintain healthy red blood cells and proper hemoglobin synthesis. If there's not enough B12, for instance, then that can create red blood cell production problems that we'll look at when we talk about anemias. Same thing with folate. Okay, so this is figure 20-15 in the book, and this is the iron cycle. So let's start at the beginning. Red blood cells are made in the bone marrow, and when they're mature, they'll go out to the bloodstream and do their job for about 120 days, and then they get recycled. So macrophages of the MPS in the spleen or the liver will break down those red blood cells. And because this is the iron cycle, let's follow where iron goes. So as red blood cells are being broken down, hemoglobin gets broken down as well. The globin chains get broken down, the heme discs get removed, iron gets removed from the heme discs, and then what we're left with gets metabolized into bilirubin, which gets secreted by the liver and the kidneys. But let's look at iron here. So now we've got this free iron. Iron can be stored in the liver. Iron could be stored in the spleen. Or iron could be delivered right back to the bone marrow to get incorporated into new red blood cells and their new hemoglobin structures. So the body is really efficient in recycling iron. Remember all the math that maybe somebody will do, I'm not going to do it right now, of how much iron is needed and required in the average adult human to maintain those numbers of hemoglobin per red blood cell. So it's a vitally important mineral for the body, and under normal circumstances, the human body is really efficient at recycling iron. We don't really get rid of a whole lot of iron at all. We just recycle it back into new red blood cells. So then let's look at what happens with bilirubin. So red blood cells produced in the bone marrow live 120 days in the blood supply and then get broken down and recycled by macrophages in the MPS. Globin gets broken down into its amino acids, and then the heme discs, remember, iron gets removed from the heme, and then what we're left with is metabolized into bilirubin. And then bilirubin gets conjugated and soluble within the liver and is excreted by way of the bile from the gallbladder, or we urinate that extra bilirubin out by way of the kidneys. So what is the condition called when there's too much bilirubin floating around? Hopefully you're thinking of jaundice, and you would be right. So what color is associated with jaundice? And if you're familiar with jaundice, you remember that it's kind of a yellowish color to the skin and especially the sclera of the eyes. So if there's a problem with maybe there's too many red blood cells getting broken down, or maybe the liver can't metabolize bilirubin effectively, then that bilirubin is going to increase in concentration in the blood and deposit in the soft tissues like the skin and sclera of the eye. And bilirubin is yellowish in color, and even sometimes yellow-greenish. And that's one of the reasons why bile has the greenish appearance that it does, is because we've got metabolized bilirubin in the bile that gets excreted by way of the feces in the gut. Okay, so let's take a break now. This will be the end of part 1 of the normal hematologic structure and function. In the next part, we're going to look at thrombopoiesis and specifically the clotting cascade and what's going on with coagulation and hemostasis. So stay tuned for that.